I'd like to first thank the College of DuPage and Bruce Seawick for hosting and organizing this afternoon, this educational event. I think the college especially is pretty open-minded about a topic that has generated controversy in the past, and as Bruce said, I think there's a renaissance occurring if we look at the scientific literature and clinical studies that are ongoing, and the College of DuPage is certainly on the forefront in this regard. I'm going to talk today about, well, psychedelics. I'll introduce them. The title of my talk is Science and Spirit. Before I begin, I just want to acknowledge the contribution of Dr. Natalie Metz, who contributed significantly to the material, the content that I'm going to show you today. I'm going to kind of begin with an introduction to psychedelics, name a few of them, and discuss their basic pharmacology. And then I'm going to kind of focus on a historical treatment of how psychedelics helped shape modern psychopharmacology and our understanding of how the brain works in a biochemical fashion. I'm going to then kind of emphasize some work that's been done with dimethyltryptamine, or DMT, a powerful psychedelic substance that's found throughout nature and including in our own brains. And then kind of give a summary of some of the current clinical research that's ongoing as we speak. So first few slides here are just kind of slides to get us thinking, what is science? What do we mean when we talk about science? What does science mean? Anybody have any ideas? Yes?
The study of how things work and why they work the way they do.
How things work and why they work the way they do. That's true. Anything else? Yes?
It's tied to the method that's used to investigate and prove, and give evidence. And it's very tied to that.
So science is tied to a method that gathers evidence, and in fact is called the scientific method. Formulation of a hypothesis, experiments, data analysis. Anything else? Yes?
Science is a subject I hated in high school.
A subject you hated in high school. You're out of high school now, and hopefully your interest can be regenerated in it. It also, it has predictive power. We're able to, model the world and make predictions about events that haven't happened yet. How about spirit, which really comes from the word for breath? When we think of spirit, what do we think of?
Inner self.
Pardon?
Inner self.
Inner self. Okay. I see we have a room full of scientists. Where are the spiritualists?
Maybe just the life that goes through you. It's the life force.
The life force that goes through us, spirit. Yes?
Yeah, animating energy.
Animating energy, okay. So things that are kind of maybe not necessarily amenable to experiment. We sometimes try, but it's a little bit maybe not as rooted in physicality. Just some other ideas. We started at some point in the past, as this cartoon says, like all other life forms today, man started as a single-cell organism in the primordial soup. And so the idea that we started as some physical events that happened in the ancient earth, and perhaps spirit came along. Here on this slide, we see like molecules. This is the primordial soup and an evolution through single-cell organisms, fish, human beings, and perhaps ultimately going out beyond the planet. Here again, molecules perhaps forming DNA and evoking kind of the idea of a spiritual, perhaps, realm here. We see some haziness out here. We see stars. And here's man in the middle. Here's a DNA strand, and perhaps man is I should say humans are the intersection between science and spirit. And the study of consciousness itself is really a blend between science and spirit. In, I would say, the last 50 or 60 years, science has been employed in trying to understand spirit, human consciousness. So this will be an introduction to psychedelics. We'll look at some specific drugs and how they were developed. The major psychedelic drugs are listed on this slide here. We have LSD, psilocybin, mescaline, and dimethyltryptamine, or DMT. Now, the first substance, LSD, is a synthetic substance, but the other three substances in that list are all natural products. So they're either found in the plant world or, in the case of DMT, are found in humans and other animals as well. The person shown on this slide is Albert Hofmann. At the age of about 100 years old, he was the person who discovered LSD. He synthesized this molecule in 1938. It was unremarkable. The pharmacologist in his group didn't see anything special with it. And he worked for a company called Sandoz in Switzerland. And so he put it back on the shelf. And then about 5 years later, for some reason, and he states here, LSD wanted to tell me something. He had a notion that he should resynthesize this material and test it again. So he did in 1943. And this is the chemical structure of LSD. And he accidentally ingested some after he synthesized it and had a kind of an altered state lasting a few hours. This was on April 16th, 1943. And so he went home and eventually got settled and tried to understand, well, what had caused this kind of psychic changes that he experienced. So he went back to the lab on April 19th and took a purposeful ingestion of LSD-25. And he took what he thought was a small dose, a quarter of a milligram. Now, there are really very few drugs that are active under a milligram, if not tens of milligrams. So he thought he was being conservative. Well, it turns out he took approximately what we would now consider about 2.5 doses, 250 micrograms, a quarter of a milligram, and had a profound experience. Bicycled home. The use of cars was discouraged. It was in the middle of World War II, and gas was in short supply, so his assistant bicycled home with him, and this gave meaning to the original Bicycle Day, which Professor Roberts actually articulated, and it's now celebrated around the nation. People send postcards to each other, greeting cards on Bicycle Day. But it's meant to commemorate the discovery, the remarkable discovery that a quarter of a milligram of some substance could produce profound mental changes, and in a way that kind of mimicked what was already known about mescaline, but far more potent, 1,000 times more potent. Yeah. So this is just a picture of some LSD. This is Sandoz LSD, and these are LSD crystals. And here's a picture of Albert Hofmann, painted by Alex Gray. He described this painting as, this here is an angel crying tears of compassion and understanding into the LSD molecule being held by Albert Hofmann. The word psychedelic itself means mind manifesting. And this term was coined by Humphrey Osmond based on a dialogue he was having with Aldous Huxley. So here's Aldous Huxley, here's Humphrey Osmond. Huxley, of course, is a famous author. Osmond was a scientist working in Saskatchewan, Canada, and they were having a dialogue and seeking a name for these kind of substances. In a letter to Osmond, Huxley wrote, "To make this mundane world sublime, take half a gram of phanerothym," phanerothym being something that induces dreams. To which Osmond responded, "To fathom hell or soar angelic, just take a pinch of psychedelic." And that was the first use of this word, and it's a word that has stuck to the present day. Some features of psychedelic experiences are listed on this slide here. There's an alteration of familiar mental frameworks, and these include things like altered sensory perception, the phenomenon of synesthesia. Has anybody heard of synesthesia before? Anybody care to elucidate? I see a lot of yes.
It's like when you have kind of cross-sensory perception, like you've experienced sounds as colors and things like that.
Yes, precisely. It's the perception in one sensory modality of an input in another sensory modality, seeing sounds, tasting colors, etc. Altered time and space perception. So often there's time dilation where a period of a few minutes may be perceived as taking a few hours or even days in subjective time. Changes in body image and sensation and a dissolution of ego boundaries. So people report kind of feeling one with the universe, feeling connected with all life forms. And this is a common occurrence in psychedelic experiences. And there are also intense mood changes, which can range from euphoria to sadness to joy to humor to sometimes even panic. A sense of profound meaning or insight, feelings of wonder, awe, beauty, and spirituality. And in fact, as we'll see, some of these substances are precisely used for their spiritual practices among native peoples and even among modern peoples. Set and setting are two qualities or two elements that were recognized early on to be important to the quality of a psychedelic experience. Set refers to a person's mental expectations, mental ambiance. How are things in your life? Are you happy? Are you dealing with some crisis? That's mental set. And then setting refers to the physical environment in which a psychedelic substance is taken. And it was discovered that both of these together had an important influence on the kind of experience that a person would have. These substances are not inherently toxic. There are a couple substances, such as ayahuasca and peyote, which do produce nausea and vomiting in some persons, but this is not considered really a toxic effect. In fact, people that use these materials consider this a purging of toxins from the body and essentially welcome those properties. Some uses are for shamanic or religious purposes. As we'll see, especially with DMT, there's evidence that this substance was used at least 3,000 years ago for shamanic purposes. For sensory or aesthetic enhancement, self-exploration, insight, interpersonal and transpersonal awareness. And this kind of relates to the dissolution of ego boundaries. So persons can have experiences, kind of a group consciousness. They report that kind of feeling. They also may bring awareness of shadow aspects of the psyche forward. So people may experience parts of themselves that maybe they're not happy with, or parts that they want to improve or change, what we might call a shadow aspect. Some persons also report paranormal experiences, such as telepathy, precognition of future events, and synchronicity. There are also, especially more recently, therapeutic and psychological healing are two aspects of, or uses for psychedelic substances. They were initially tried in this regard in the 1960s. Some of this research was ongoing when research with these substances was prohibited. However, recently have reemerged in this context, especially with persons who are dying of terminal cancer, and also for psychological healing in terms of addictions. There's been some very promising work recently with people who have addictions, both to opioids, alcohol, nicotine, and also research into consciousness. And we'll talk about this shortly, how psychedelics really form the seed for our current understanding of how neurochemistry works, how brain chemicals influence mood and memory and cognition. Importantly, it's good to understand what conditions will sustain the use of psychedelics in a healthy and beneficial way. So in ways that promote healing, that promote community. So set and setting, again, are two important parameters. Also the substance and the dose that's taken. These substances, we kind of put them under the umbrella of psychedelic, but to be sure, they all have unique effects and they're all discernible among people. And then the size of the dose has an influence. It's been increasingly acknowledged that a guide or a sitter is important to help shape the experience and to help the person having the experience have a healthy experience. If a person is going through a tough time or they're dealing with some inner conflicts or past traumas, then having a guide or sitter present can help a person work through those traumatic events. Nutritional status and physical condition, of course, will influence what kind of experience a person has, so it's important to have sound nutrition, to be physically fit, to be emotionally settled, to be happy in a person's life. Psychosocial factors, do people have pre-existing psychiatric problems, familial history of psychiatric problems, and also these experiences there should be some mechanism whereby they can be integrated into a person's life. We see that with native societies, for example, because these experiences are part of the culture, it's easier for these people to integrate the experience that they have in a peyote meeting, for example, into their life, into their community. I think one deficit that happened in the 1960s is all of a sudden we had access to these materials, but there was no context in which to put them in, and so that it was hard to integrate these experiences into Western society, and so there was there was a disconnect between these experiences and the way our society functioned. To be sure, these substances are not entirely without adverse effects. Adverse effects can occur. The most common adverse reaction, which still occurs quite rarely, 0.1% of the time, is an acute panic reaction, a so-called bad trip. This typically lasts less than 24 hours. But it's about 1 in 1,000 people will have that kind of experience. Basically, a confused state, paranoia, anxiety. Now, there are some things that correlate with that state, which can essentially favor having that state. And so these are some of the correlates. Drug taking alone or in an unstructured setting, and so this is the affective setting. Excessive initial apprehension. If a person isn't prepared for the profound changes that may occur, that can predispose them to being confused or anxious. Rigid and brittle psychological defense structures. Considerable subsurface guilt and conflict. And submission to peer pressure as a motive for drug use. So if one avoids this set of conditions here, one will further decrease the potential to produce a panicked or anxious state. The best treatment is a non-drug treatment, but it's simply reassurance, so-called talking through the experience. So a person can be, who's undergoing a panic reaction, can be taken into a quiet environment, reassured that the drug effect will wear off shortly, that everything is fine, that they're going to be fine, and this generally has beneficial effects on them. In some persons, even more rarely, this is 8 in 10,000, psychosis lasting more than 48 hours. This is correlated with a history of psychiatric illness or treatment or a history of polydrug abuse. So persons in current studies are screened for these conditions. And so these would be exclusionary criteria. Persons who have had a history of psychiatric illness or treatment would probably not be allowed in one of the current psilocybin studies, for example, or a history of polydrug abuse. As I mentioned earlier, nausea and vomiting are common with peyote and ayahuasca, but these are not considered adverse effects. These are considered beneficial by the persons taking those. Brief word about indigenous entheogens. I'll bring up the word entheogens here. Entheogen means to generate the God within or generate the divine within. It's derived from Greek meaning entheos, God within, genesthe, to generate. And so this is the use of psychedelic agents really for a specific purpose, really for spiritual purposes, not necessarily for healing, although certainly spiritual experiences can be psychologically beneficial. But these are the use of these substances specifically to contact spirit. This is just a painting that kind of brings together the use of plant materials by native peoples. You can see how these people are actually composed of leaves and plants. Tuvan shamans who use some of these agents here having a ceremony around a structure that they've created. And so humans have used psychoactive plants and fungi for millennia, and this is just an example of some of them. Banisteriopsis caapi, this is a vine that's used to make ayahuasca. Amanita muscaria, a so-called fly agaric mushroom, which has some psychoactive components in them, not normally considered psychedelic, but certainly have been used for many centuries by people for the psychoactive state it produces. Psilocybin mushrooms, which contain the active ingredient psilocybin, which indeed is a true psychedelic, and Lophophora williamsi, the peyote cactus, which has been used for centuries by native peoples, the Huichol in Mexico and others in southwestern United States. This is just a map of the world showing some names and pictures of psychoactive plants around the world. And so you can see that they're really found throughout the world. These are very, actually quite commonly found. And so just some pictures. This is Psychotria viridis, datura type plant, etc. And the artwork that has been produced by some of these native peoples, this is the Shibo people. They have kind of a characteristic form of artwork. And these essentially are songs that have been visualized, put into visual form. These people, the shaman, when taking ayahuasca, will sing a song into the person who they intend to heal. And when they see this pattern settle onto the person, then they consider that the song has been effective. But essentially what these are like a visual representation of a specific song that the shaman will sing. But beautiful artwork. Here's some artwork inspired by peyote. This is a Huichol yarn art, a specific form of art found in Mexico. So very colorful. Here's a peyote cactus right here There's a couple other ones up here, but very bright and kind of evocative of the psychedelic state, in which colors can be very much brighter than normal. This is trying to evoke some visual interest in a non-psychedelic form, but if you can see the screen kind of wavering, it represents what some people have reported under the influence of psychedelics, that surfaces may shimmer and wave. So I'd like to now kind of get a little bit more into the hardcore science part of this and discuss neurochemistry, psychedelics, neurochemistry, and mental state and the relationship among them. And so if we begin with a sober brain, a psychedelic brain or a high brain is this side, the right side, lots of colors breaking through the barriers. And our understanding now of where psychedelics work is really at the synapse of the neuron. So this is just a diagram of nerve cells in the brain. And the synapse is the point where two neurons almost touch each other. They don't quite touch. There's a gap there, and that's called a synapse. And we now understand that neurotransmitter is released. These are substances like dopamine, serotonin, norepinephrine, other neurotransmitters, histamine. And they then act upon receptors on the target cell. Now, this is kind of common knowledge now. I think everybody in this room has probably heard something like this, but I would rewind the clock back to about 1950. And at that time, it was believed that brain function was entirely electrical in nature. There was no conception that brain chemistry had any influence on moods or memories or behaviors. And it was really, we'll walk through how psychedelic drugs actually changed that and contributed significantly to this kind of picture, which we now take for granted. So this is serotonin, the chemical structure for serotonin. It was known from the late 1860s that if one obtained clotted blood, that there was a substance in the liquid, not in the clot itself, but if you centrifuged that and got rid of the clot, you took that liquid and put it on a blood vessel, it produced vasoconstriction. And they called this hypothetical substance vasotonin. We now know that it was, in fact, it's serotonin. The chemical structure was identified in the late 1940s, and the chemical synthesis of the pure compound was done in the early 1950s. So 1951, serotonin became available to researchers for experimental purposes. And the person that really connected serotonin with the effects of LSD and mental state is this guy, Sir John Henry Gattam, a famous British pharmacologist. He studied the gut, and he discovered some important peptides, such as substance P, peptide that signals pain signals. But this is John Gattam. And he did some self-experiments with LSD, and it just so happened he was also studying serotonin. But before that occurred, in the early 1950s, he was one of the players, but Betty Twerog and Irving Page in the United States, and Gattam is over in England, showed for the first time that this substance serotonin, which had just been synthesized, was found in the brain. In fact, Betty Twerog developed a very sensitive assay involving muscles like clams, those kind of muscles. The muscle's muscle. It's kind of funny, I guess. But that would respond, would contract if serotonin was applied. So people started looking at all sorts of tissues and spritzing some on these muscle preps, and lo and behold, they found it in the brain. Gaddam had taken LSD on 4 occasions in the early 1950s, and he reported kind of the common things that we associate with psychedelic experiences. I don't know about a feeling of irresponsibility, but certainly euphoria, increased awareness of shapes and colors, shimmering of peripheral vision, and a dreamy feeling that sensations did not represent real objects. Because he was studying serotonin, and I'll show you how the chemical structures overlap, he applied LSD to his tissue preps of serotonin. He discovered that LSD antagonized the effect that serotonin had on tissue. And so that was published in this paper in 1954. And he hypothesized that the psychoactive or psychedelic effects of LSD resulted from its actions on serotonin in another paper in 1954. So this is really the first kind of inklings that chemistry could affect, or in particular brain chemistry, could affect mood and perception and memories, etc. The first statements linking the effect of LSD to serotonin were by Gaddam, and I quote directly from this paper: the evidence for the presence of HT, which is serotonin hydroxytryptamine, in certain parts of the brain may be used to support the theory that the mental effects of lysergic acid diethylamide are due to interference with the normal action of this HT. Lysergic acid diethylamide, of course, being LSD itself. So this was directly out of this paper. Simultaneously, Woolley and Shaw, 2 American pharmacologists, published another paper in 1954. So these are 2 completely independent groups. I mean, talk about like synchronicity, that they were on the same path. But in their paper, they said that the mental disturbances caused by lysergic acid diethylamide were to be attributed to an interference with the action of serotonin in the brain. Gaddam also was cognizant of the mental effects of lysergic acid diethylamide and of the occurrence of serotonin in the brain. We have surmised that he has been thinking, just as we have, about the relationship of serotonin to the mental disturbances induced by the drug. So this was a huge quantum leap, essentially, in understanding how consciousness occurs and is shaped by chemicals in the brain, in particular serotonin, but that you could administer other agents that would interfere with this. And so now we had a tool that helped us understand how consciousness is shaped in the form of particular LSD. I'm going to shift a little bit now to dimethyltryptamine, N,N-dimethyltryptamine, talk a little about that, and you will see how this converges with LSD. So dimethyltryptamine, the structure is shown here, is a very potent psychedelic agent. It was first synthesized in 1931 by Canadian Richard Mansky. Didn't do anything with it. It was just part of a chemistry project he was working on. In the mid-1950s, this substance was identified in hallucinogenic snuffs that were used by certain native peoples in Haiti and South America. These snuffs are called cahoba or yopo. And so this substance was identified in the snuff. So we had like another piece of the puzzle there. It's like, well, you know, it led people to that question, like, well, maybe this stuff is what's causing the snuff to be active. Furthermore, when I first started, I mentioned that DMT has been used for at least 3,000 years. So there were a discovery of these pipes. These are prehistoric pipes dating to about 3,000 years ago in South America. They had traces of plants that contain high amounts of DMT in a pipe to smoke. Now, we now know that DMT must be ingested either by smoking or parenterally. If you take DMT by itself orally, it's completely inactive. And so this kind of fits with the use of a pipe to smoke this plant material. It's a major component of ayahuasca tea used by the Santo Daime Church and the União do Vegetal. Now, I just said that if you take it orally, it's inactive, but this tea is certainly active. Well, the reason is the tea contains another plant, the Banisteriopsis caapi I showed you earlier, that contains substances that allow the DMT to be orally active. If one just takes DMT by itself, there are enzymes in the gut that will degrade it very rapidly, and you can never ingest enough to have the psychedelic experience. But with this Banisteriopsis caapi, it contains naturally occurring substances that block these enzymes. And so by blocking these enzymes, the DMT scoots through, gets into the circulation, and then produces the psychoactive effects. Enter Stephen Zara, a Hungarian medical doctor. Zara was the first person to discover that DMT itself had psychedelic effects. Now, as I just said, people have been using these substances for centuries or millennia, but they didn't know that it was the DMT that was producing the effect like an ayahuasca tea. But Stephen Zara obtained pure DMT, and he discovered that DMT produced psychedelic effects similar to those produced by mescaline and the already, at this time, known LSD. He discovered that it was inactive orally, so when he took it, it did nothing. But when he gave it to himself intramuscularly, it produced visual illusions and hallucinations, distortion of body image, speech disturbances, and mood changes reminiscent of those produced by LSD or mescaline. The effects had a rapid onset, 2 to 5 minutes, and a short duration of action, being entirely dissipated within 60 minutes. And he wrote about these discoveries in this article in 1956. So you see what's happening between 1951 and 1956. Like all this information about neurochemistry and these psychoactive drugs influencing serotonin, and they produce intense mood changes, and so interest was piqued in a biochemical explanation for consciousness. I mean, if you just have studied psychology, you know that behaviorism was like all the rage back then, B.F. Skinner, et cetera, and there was really no consideration that brain chemistry had any influence on an animal's behavior. The Canadian group, in fact, this is Humphrey Osmond, who we used his quote before, hypothesized that the body could produce an endogenous substance, which they called a schizotoxin. Under certain conditions, aberrant metabolism might produce some substance that generated the mental disturbances seen with schizophrenia. And they published in these two papers here. 52 was the first articulation. They proposed that something related to adrenaline metabolism, but even they said that they couldn't imagine any kind of metabolism that would produce sufficient, that would convert adrenaline to mescaline, for example, in sufficient amounts to produce the effect. So they proposed this, but they didn't really have anything in mind. They called it M substance at the time. And then in 54, reiterated that. So this was the first idea that proposed to link mental disease to aberrant neurochemistry. I mean, now this is kind of accepted. It's like gold standard. Drug discovery by pharmaceutical companies is totally geared to treating mental diseases by affecting the neurochemistry. I mean, the whole billion-dollar industry of things like Prozac and, olanzapine and Strattera and all these substances can be traced directly back to these early studies on neurochemistry related to psychedelics. So this just compares the structures of serotonin, DMT, and LSD, and you can see the pink part of the molecule here overlaps. And so these people looked at these substances and these structures and said, "Oh, now I know why it's working. It's mimicking serotonin. It looks chemically like serotonin." And so that was how that kind of came about. The picture now moves ahead to Julie Axelrod. He's a Nobel laureate. He won a Nobel Prize for his work in neurochemistry and synaptic vesicle release of neurotransmitter, working in his lab. But he made the discovery in 1961 that animal tissue, he used rabbit lung in this experiment here, could convert tryptamine. So tryptamine is a substance obtained from the amino acid tryptophan, which I'm sure everybody's heard about tryptophan, right? Like stuff in turkey makes you sleepy on Thanksgiving, you know. But so tryptamine comes from tryptophan. He demonstrated that if you took tryptamine and incubated it with animal tissue, you could form DMT, would pop out the other end. So now this theory of the Canadians of a schizotoxin all of a sudden had teeth, because he demonstrated that you could produce DMT in an animal tissue. I mean, we already knew you couldn't produce LSD in animals, and again, mescaline would have to be produced in such high amounts that it would be kind of unreasonable, but DMT is quite potent. In 1972, Axelrod and his co-worker Juan Saavedra showed that human brain tissue could carry out the same transformation. It's like, wow, now we're really kind of getting somewhere. If human brain tissue can create DMT, you know, maybe that explains certain mental states. And so this is the transformation of tryptamine to DMT. For the non-chemists, this is tryptamine formed from tryptophan, the amino acid tryptophan. It's found in all of our bodies. And there's a two-step mechanism that adds a methyl group to get dimethyltryptamine. These are the two methyl groups. And the methyls come from this molecule S-adenosyl methionine, also known as SAMe. It's a big seller at health food stores because of its wide range of effect on other biochemistry. But there was an enzyme, indolethylamine N-methyltransferase. Try saying that 3 times while standing on your head. It's kind of a tongue twister. But it wasn't, the enzyme itself wasn't known when Axelrod first did his experiments. It was hypothetical. It was like there must be some enzyme in the body in an animal tissue that can carry this out. But it was purely speculative. And for the next 25, yes?
Going back a little, when you showed how the chemical compounds have a similar base that from serotonin to LSD or DMT, are you saying that so they get that part gets recognized and it blocks it goes into the receptors? Is that what you're saying? Because are you saying that they antagonize the receptors for serotonin, that they're antagonists, or I was just confused about what you were saying.
You're exactly right up to a point. So we now know that they're what are called partial agonists. So they don't actually block. So in the presence of serotonin, they inhibit the effect of serotonin. But if you don't have any serotonin present, like in a tissue prep or something, and you add these things, they mimic serotonin exactly. And they turn on the receptors, but only partially. They're not, serotonin is what's considered to be a full agonist. So whatever it does to the receptors, it does it at 100%. Drugs like LSD and DMT are partial agonists, so they turn on the receptors, but only to the tune of maybe 30 to 50%, okay? But you're exactly right. It's that part of the chemical structure is like a lock and key. It's recognized by the receptor, so the receptor thinks it got serotonin when really, you know, it's LSD that's binding and tweaking the receptor. In fact, you can think of a full agonist as a key that always opens the lock 100% of the time. A partial agonist would be a drug that fits the lock, but you have to jiggle it, and then sometimes the lock will pop open. So it's not as effective as serotonin by itself, but they do in fact open the locks. Does that make sense? Okay. So for about the next, after that 72 paper by Axelrod, for about the next 25 or 30 years, people tried to see whether, does human brain make DMT? Some people found DMT in human blood, in human urine. Others were unable to replicate those results. So there was quite a bit of controversy until about the late 1990s. Enter Mike Thompson. This guy cloned the enzyme that was hypothetical. Remember I showed you, I said this is completely hypothetical, the INMT, indole ethylamine and methyltransferase. This guy cloned the enzyme and reported about it in 2 papers in 98 and 99. He discovered the gene itself and its chromosome location. So now, beyond a shadow of a doubt, we all carry the gene that makes INMT. So we have the hardware to make it. I'd like to talk briefly about some work I've been involved in, because there's some really cool pictures I want to show you at UW-Madison. So that's my home turf. And so we've been working on tryptamine and INMT at the UW. We've published on DMT and sigma-1 receptors. These are our publications. DMT and serotonin uptake transporters. Various new ways to synthesize tryptamines, including microwave-assisted synthesis, which turns an overnight reduction into a 5-minute process. In particular, the identification of the enzyme, the INMT enzyme in primate tissue. So when Thompson cloned that gene, okay, they discovered the gene, but they didn't actually show that the protein was present in nervous system tissue. And so we were able to obtain antibodies from Thompson and use this kind of assay. I just want to set this up for you so when you see the pictures, you'll appreciate what you're seeing. So this is a common, it's called immunohistochemistry, and it's used hundreds of times every day across the country to look at proteins in various tissues. This is how it works. If we have a tissue that contains the INMT, this enzyme that makes DMT, present, then it's possible to generate an antibody against that protein. So, and this is what we were able to obtain from Mike Thompson. This is an antibody generated in rabbits that recognizes this enzyme, INMT. So it sees it, basically the rabbits were injected with with human INMT protein, and they saw it as a foreign protein. Your immune system's kicked in. It's like, oh, I've got an infection, so they make antibodies against it. So it's possible to then take their blood and harvest the antibodies from it. And so now you've got antibodies generated in a rabbit that recognize primate INMT. Then commercially, you can buy these goat anti-rabbit secondary antibodies. So now you have what you've generated in a goat is proteins that recognize rabbit proteins as being foreign. Got it? Okay. And these are commercially available and they're tagged with what's called a fluorophore. So this is a molecule that if you irradiate it with light of the right frequency, it'll fluoresce, in particular green. So wherever you see green on the upcoming slides is going to be INMT. You just got to follow the chain down. Now consider, so if you incubate these antibodies with tissue that has INMT, it'll stick to the INMT. If you have tissue that doesn't have INMT, then there's no place for the antibody to glom onto, and you can just wash it away, and now when you go look, there won't be any signal, it'll just be dark. And so it's easy to see where the protein is and where it isn't. So first we looked at 3 nervous tissues. We looked at spinal cord, retina, and pineal gland. So this is spinal cord motor neurons. We're kind of looking down on the spinal cord now, so this green thing here is a motor neuron that innervates your skeletal muscles, okay? And so this is a photomicrograph of spinal cord motor neurons. Wherever you see green is, in fact, that enzyme INMT that makes dimethyltryptamine or DMT. The blue staining are nuclei of other neurons, so it's just a stain for nuclear DNA, chromosomes. And you can see immediately that the green isn't all over the place, it's specifically located. And so that gives us confidence that what we're seeing is a real signal. We also kind of zoomed in here, and so we looked to see if the INMT co-localized with the sigma receptor. So this is a neuron, a terminal of a neuron. Green here is sigma-1. Chat is an enzyme that makes another transmitter, acetylcholine. You don't have to worry about that. We're going to really look at the green signal. So this is green on this side is sigma-1 receptor. Green on this side is INMT. And so what we're seeing here is that they kind of overlay on top of each other. You see they're in the same location. So they're physically close to each other in space. This is primate retina. So this is a cross-section of a retina. And so these are the rods and cones at the bottom. There's a number of other cell layers. Light comes in from the top, goes through several cell layers, and then hits the rods and cones. And they activate, and you see. And so wherever you see green here is INMT. The blue here are these nuclei for this so-called outer nuclear layer. The reddish-yellow area here where synapses form. Here's some more INMT. Here's the inner nuclear layer, these blue nuclei here. And then some more synapses, and then the green here are ganglion cells. I will say that the red, synaptotagmin 1, just FYI, is a protein that's found in synaptic vesicles. Is everybody okay with synaptic vesicles? Have you ever heard that term? Well, for those who haven't, these are small organelles within cells that contain the neurotransmitter. They contain serotonin or dopamine or norepinephrine. And so it's these vesicles that fuse and release the transmitter. So they all contain synaptotagmin 1. So what we're seeing here is just a marker. It's just an indicator that there are vesicles there. That's all that the red means. But so the ganglion cells, the green ones here, these are the cells that actually form the optic nerve. So now we see that several places in the retina is the protein that makes DMT, the smoking gun, if you like. And then finally the pineal. The pineal is buried deep in the brain. This is labeling of primate. So all the green that you see is the enzyme INMT that makes DMT. Again, the blue is nuclear protein. The red is a synaptic vesicle. Another view of the pineal gland. All the green spots are INMT. Now, as I say, it's the smoking gun. It's not DMT itself. But Rick Strassman, who's been very interested in this area for a number of years, recently did some work that showed in real time in rodents that they were actually producing DMT in the pineal gland. So his work with DMT is he was the first one to conduct human studies in the mid-1990s with a psychedelic agent. In particular, he used DMT. There are a number of reasons, partly due to its short activity, partly due to the history of it. We knew about it. It wasn't some new drug, and it was a natural product. So he administered several hundred doses to about 60 volunteers. He wrote about his research in DMT, the spirit molecule, and this is the cover to that book, and it was made into a movie in 2010. He's currently working in New Mexico as a clinical associate professor of psychiatry. So he's been speculating about the role of DMT in consciousness for quite a while. He proposed that maybe, he and others proposed that DMT might be involved in dreams, might be involved in near-death experiences, might be involved in spontaneous spiritual experiences. His latest work was, as I mentioned, he demonstrated in real time that rodents, rats, produce DMT. I'm just going to briefly walk you through this, and then we'll talk about some current clinical research and then wrap up. So what he uses a technique called in vivo microdialysis. So what this is a probe that can be inserted into tissue, in particular into brain tissue, and one can pump in a fluid, and it goes down into this probe, and then it flows out, and it goes out this way. And at the very tip of the probe, there's a dialysis membrane. So this is a semi-permeable membrane that blocks the passage of large molecules, but permits the passage of small molecules, for example, DMT, across the membrane. So it's kind of like a very fine mesh. You could think of it that way. And so what occurs in a microdialysis experiment is the fluid is pumped in, it goes down the central shaft, and then it goes out. See these red arrows? And then it goes out the outlet. And anything that is in the tissue can be taken up by the probe. In fact, there's a driving force, a concentration gradient that will drive these substances into the probe. And so if, for example, DMT is produced, you can see that by analyzing the fluid that comes out the outlet. And this is actually their data that showed that. On the right panel is authentic DMT. So they just made some DMT, put it in solution, and looked to see where it would elute. And they used a technique called mass spectroscopy, but we won't get into that. If you just believe me that these peaks represent authentic DMT, and then these first three here, these first three columns are effluent from a rat pineal gland. And you can see a number of other compounds here in the rat, but they all share the same peak here for DMT. And what you see here are these numbers are molecular weights. And so it's just that they share, it kind of gives confidence that what you see that comes out of the rat is in fact DMT. So that was just published this year, about 6 months ago. So it's very exciting. For the first time, they've demonstrated that a living mammal can produce this psychedelic substance in vivo. All right, so this is DMT, the colorful, the spirit molecule. Again, Alex Gray seems to be a darling of the psychedelic community. He's got a very characteristic form of art that he does, but it's certainly a colorful molecule. And we've taken it from its use 3,000 years ago to its discovery that it was active. It was found in various snuffs. And then the story going up from the 60s, 70s, 90s about how it very much drove and influenced our current understanding of brain function. And so we don't know where it's going to go from here. But there is some current clinical research, and I will finish up with this. I just want to give you a brief overview of what's going on in the present day. So these are some studies, and I borrow this title from Tom's book, Psychedelic Substances as Treatments. These are some medical uses for psychedelic agents presently. So there are a couple groups at these University of Arizona, UT, San Antonio, using psilocybin to treat obsessive compulsive disorder. Psychedelic therapy for the treatment of depression, Michael Montaigne at Massachusetts College of Pharmacy. Response of cluster headaches to psilocybin and LSD, Torsten Passy in Hanover, Germany. John Halpern at Harvard. And I put a link to the Cluster Busters website. So it's been found that these agents can ameliorate cluster headaches. They're called cluster headaches because they occur in clusters in time. So a person who has this condition will experience many headaches, maybe over a period of a day or a couple days, ice pick type headaches, very excruciating headaches, debilitating headaches. They might occur for a few days or a week, and then they don't occur again for maybe 6 months, and then all of a sudden there'll be another cluster. And it was discovered that these agents, psilocybin and LSD, and now a non-hallucinogenic chemical cousin of LSD called BOL 148 has efficacy in treating cluster headaches. And there's a great deal of interest in this presently, and some studies are ongoing with this. Also, and this is from the New York Times about about a year and a half ago, the use of psychedelic drugs to dissipate end-of-life anxiety in terminal cancer patients. So this is work being done at Johns Hopkins University, at New York University, and these are the groups here, and also at UCLA. So use of psilocybin in patients with advanced cancer, Charlie Grobe, Roland Griffiths at Johns Hopkins, and Jeff Gus and Steve Ross at NYU, and here are links to their websites. These slides will be available for people who are interested afterwards, so you can follow these links if you have further interest in it. But these studies are actively recruiting patients who might benefit from this. Hallucinogens in the treatment of alcoholism and other addictions, John Halpern at Harvard, Alper at New York University, and I will also now mention Bogenschutz, University of New Mexico, is doing some very new work using psilocybin to help treat alcoholism. And then MDMA, I don't really personally consider it to be psychedelic like the classic psychedelic agents. It has unique effects on emotions. Some people do lump it in together, but Mike Mithofer at South Carolina is using MDMA to treat post-traumatic stress disorder, and it has had some very good results with it, with MDMA. These are some professional and popular publications in recent years regarding psychedelics. The Lancet, respected British medical journal, Scientific American, New York Times, etc. Here's some other links for you to check out for resources, in particular students who are interested. There's a site at the MAPS site that is of particular interest to students, how you might, if you're interested in pursuing this, some approaches that you might take, some resources there. So with that, I will say thank you, and I'll entertain any questions you might have. Yes?
I was surprised to see the research that you identified in depression, because you had earlier said that some of these drugs have problematic issues, you know, if you've had a history of mental illness.
Yes, right. I think yes, a great question and very astute. The main concern is probably schizophrenia and psychosis rather than depression per se. Some have found, in particular the use of psilocybin by the group in Arizona, showed promising results in people that were treated with low doses of psilocybin in treating depression. And so I'm not a psychiatrist, but I've looked at their data, and they use certain instruments that are used to measure depression, clinical depression, and there were improvements in all the indices. So I would say that the caveat against their use in psychiatric patients probably applies more to psychosis and schizophrenia. Yes?
Since they use a lot of serotonergic drugs for depression, when you use a psychedelic, what are you doing there? I mean, because it's a partial antagonist, so I'm a little confused how that.
Partial agonist.
I mean agonist.
Right. So, well, the antidepressants, the currently used antidepressants, your Paxils, Prozac, Zoloft, Effexors, those all increase serotonin function in the synapse. So to the extent that these agents mimic serotonin, albeit only partially, that kind of still fits with that model. Now, it's recently been discovered that the drug ketamine has a profound, so one thing with the traditional antidepressants is they take weeks or months to exhibit their therapeutic benefits, if at all. but it's been discovered that ketamine reverses depression in a matter of hours, a single dose, and they've found that it lasts at least several weeks. Now, I'm not totally up on the follow-up on that, but there's some evidence that it's actually a growth factor. It's BDNF, it's brain-derived neurotrophic factor, that is increased by ketamine, and coincidentally by the traditional antidepressants, albeit 4 steps upstream. And so that might explain why they take so long, but they also increase BDNF. So the link is that they kind of mimic serotonin, and so that it's still consistent with that.
If psilocybin acted as like an SSRI or something like that.
Well, it doesn't inhibit uptake. So the SSRIs are uptake inhibitors, right? So the serotonin is, with an SSRI, it's not cleared from the synapse as rapidly, and so it lingers and turns on the receptors, right? So these agents essentially are kind of mimicking serotonin at the receptors themselves. So again, it's perfectly consistent with that. Yes?
I was going to say, decades ago, there was a story of the natural occurrence of LSD and wheat ergot and how it affected a village in France when they had bad wheat and bad
The Day of St. Anthony's Fire. I have that book on my bookshelf, actually. The Day of St. Anthony's Fire. So in medieval times, if people so ergot is a fungus that grows on rye, and if people consume this fungus, not only did it produce hallucinations, but it's a potent vasoconstrictor, ergotamine is, which is in the fungus, and it would produce dry gangrene, and so people's like fingers and toes and stuff would wither away and fall off, and they call that St. Anthony's Fire. It's largely due to the ergot alkaloids. LSD itself is not found in ergot, but there are other substances that might still produce psychoactive effects if taken in high enough amounts. Yeah. Yes.
You started off by talking about spirits.
Yes.
And throughout the talk, I did not hear you say anything, the connection between spirit and the flesh as healing is concerned.
Yes.
Because one of the examples with the Christian Medical Commission set up by the World Council of Churches was that they discovered, especially in Africa, that mental problems were easily and quickly taken care of by local herbalists than in modern hospitals. And so they had to set up a Christian Medical Commission to look at mental problems, especially in tropical Africa, and to see how they could coordinate the tropical medicine with the modern medical care. And so, even up to and including today, what happens is that those who have mental problems, when they go to modern hospitals, they are referred to traditional healers, and there they get some effective results. How do you reconcile that with this?
Well, I will defer a little bit of that to probably Bruce. In fact, his theme is mystical and spiritual aspects of psychedelics. So I kind of emphasize more of the science end of it, but I mean, certainly, partly it might be culture. So we're, kind of looking at it from a Western medicine point of view, an allopathic point of view is not necessarily the same approach that a traditional healer would use. I think certainly that spiritual experiences and one's spiritual life has an effect on health. I think that spiritual experiences are beneficial, but I would, again, defer to probably Bruce to cover that topic. But I think it's a terrific question, and I just wanted to introduce the idea of spirit, and I appreciate that I only touched briefly on some of the shamanic uses, and that you're certainly right about that. I'll try to get a better mix. next time. Maybe one last question?
More and more states having medical marijuana legalized. Are these psychedelic drugs already allowed, or are they legally being used?
Yes, so the studies I referred to they're sanctioned by the FDA, DEA, Institutional Review Board. So they've all passed several levels of scrutiny. It's all being done under, legitimate conditions. I'm unaware of marijuana research that's going on that way, but I mean, we already know that marijuana has medical efficacy. Otherwise, the drug Marinol would not be a prescription drug across the country. So it's really a question of the drug delivery system, not the drug itself. So you can take Marinol if it's prescribed, but you can't smoke marijuana in many states. I think the writing's on the wall. I think that will change. It just offers other options, vaporizers, tinctures, edibles. These are all appropriate means of ingestion. And if the drug works, the drug works. And so, I mean, okay, thanks for your attention. Thank you.